record locally as well. Great.
Yes.
Awesome. Okay, so thank you so much for the opportunity, and it's really such a pleasure to follow such a wonderful presentation. I think the Gene Matcher is extraordinary, an extraordinary resource that sort of can help many scientists. So what I can tell you about is sort of what do you do with all of these genetic hits once you have them? And in particular, I can tell you about the work that we've been doing in translating genomic findings and these hits from genetic studies into actually therapeutic insights. So number one, how do we dissect? And number two, how do we manipulate the circuitry of human disease at single cell resolution? So I'm a professor in the computer science department and the artificial intelligence laboratory of MIT, and I'm also a member of the Broad Institute. And then this is our website, compbio.mit.edu. You can find there hundreds of hours of recordings of various lectures that I've given on genetics, epigenomics, transcriptomics. So I really encourage the students to visit this link, because you will find just enormous resources. Every lecture and every class that I've taught at MIT is publicly available. So for those of you who want to learn a lot more, there's a lot more information. You can also follow me on Twitter, where I post a lot of our recent papers. So I'll start with a brief introduction as to sort of what is the starting point of what we do. The starting point is genetics, and genetics has been around for thousands of years. So our ancestors from the Middle East and from across the world, in the New World, in the Old World, have been selectively breeding animals and plants for their properties. So you can turn something that's completely inedible, like teosante, into corn, that basically form the basis of nutrition for the New World. And you can turn sheep into these, wool-producing machines that basically will suffocate if we don't cut off their wool. And you can turn something that will eat you into something that can't wait for you to get home. Through the selective breeding, through phenotypic selection, which then selects for genetics. And the inheritance, even in humans, for eye color and hair color has very long been understood. But the molecular basis for how this mechanism works was not elucidated until Gregor Mendel carried out his very elegant experiments in Brno, in the current Czech Republic, to basically look at the patterns of inheritance of very discrete phenotypes, which we now call Mendelian, which was, again, the focus of the previous presentation. So basically, for these kind of phenotypes, you have this very discrete inheritance. You can either be, let's say, purple or white. And if you have a recessive allele that doesn't express, for example, the purple color, you will be white. But if you have even one copy, you will be purple. And that basically means that inheritance is discrete. It is not continuous, that you can either have 100% or 0% of a locus. The locus can either be, dominant or recessive, and can basically yield the phenotype or not yield the phenotype. So that was a dramatically different concept from this continuous variation that people had observed. Eye color just varies continuously, and hair color varies continuously, and height varies continuously in the human population. So Gregor Mendel's experiments were dramatically different by saying that there is no blending, that there's dominant recessive alleles that are, and that was the key point of Mendel, independently assorted, that different, what we now call the genes, are inherited independently in the genome. The problem is that this was very difficult to reconcile with this continuous view of human variation, and there's many other, models that were competing. And Gregor Mendel's work basically remained unopened for 50 years. It was only covered in a local newspaper, and his manuscript had been sort of sent to Darwin, and Darwin had left it unopened on his desk even after his death. So why are these views so seemingly contradictory? This was not reconciled until the beginning of the century with Ron Fisher and other statisticians that basically said that even with 5 Mendelian loci, each of which increases or decreases your height by a little bit, you can still have a seemingly continuous phenotypic variation. The concept that you can still have these two models, you can still have continuous inheritance and these discrete units underlying this, was basically finally possible. And this has manifested in 2 different modes of studying human disease. One of them is focused on Mendelian disorders, which are inherited discretely. And interestingly, Mendelian gene mapping was made possible by the deviations from the rule of independent assortment. Namely, if 2 genes are nearby, they will not be completely independently inherited. They will be partly linked through this co-localization on the genome. And this is basically what led to the revolution in Mendelian gene mapping in the 1980s. But for complex traits, which include Alzheimer's, cardiovascular disease, obesity, schizophrenia, hundreds of complex traits, which are not due to a single or a small number of genes, this basically was simply impossible to crack using these traditional approaches. And the analogy that I want to give is the difference between the traditional knockouts that people carried out in animals, which you can think of as a giant hammer breaking one of the genes in a mouse, and then asking what is the phenotype of breaking this one gene, versus the thousands of needles that are only minutely affecting the phenotype of these complex traits in the human genome. And these complex traits, which have only these needles, these very, very small effects, were not tackleable. No one could actually go after them for the vast majority of this genetic revolution that was happening in the 80s. And it wasn't until the human genome was fully sequenced and until complete variation maps were built and that we understood the haplotype nature of co-inheritance of blocks of genetic variants in the human genome that finally enabled genome-wide association studies. And what we have now recognized is that there are truly hundreds of thousands of genetic loci, hundreds of thousands of these needles that are affecting disease in extremely important ways, but also in extremely subtle ways. And with a hammer approach of, knock out one gene at a time, you don't need computation. But when you have 1,000 needles minutely affecting a system, a phenotype, this extremely complex circuitry of the human genome, then you truly need computation. And that's where my lab comes in. So basically, we start with genetic association studies. So basically, the statistics of Ron Fisher from 100 years ago is basically what statisticians are still using to understand genome-wide association studies. You basically look at every single nucleotide variant, every single nucleotide polymorphism or SNP in the genome. And then you look at its association on the y-axis with different traits. In this particular case, I'm showing you body mass index, a measure of obesity. And what you can see in these associations is that some genetic variants show an astronomical p-value. So the p-value represents the probability that this association is observed by chance using a chi-square statistical test. So you basically ask how many of the cases, how many of the controls have allele G or allele C in this particular locus. In this FTO locus, for example, it's C for the risk, T for the controls. So, and then you see a deviation from what you would expect if this was independently segregating in cases and controls. And then you can basically say, well, the probability that this would happen at chance is 1 in 10 to the 60. And, there's only 10 to the 60 atoms in the universe. So basically, if every atom in the universe was basically a chance that you would hit with a dart, this is basically the chance that one such atom in the universe is in fact responsible for this variant. And that probability has now increased even more astronomically. So even if every atom in the universe had a universe within it, the atoms in that universe would not be enough to explain this by chance. So that basically says that statistically, there's something going on in this region that is causally associated with obesity. And why do I say causally? I say causally because the vast majority of human epidemiology has been correlation-based. And the ability to now go from correlation to causation is what we need for intervention. What we need, and can I stop my screen sharing for a second? So, hi everyone, I am human, and I hope you are too. I feel like I'm speaking to an empty wall. Could I ask everyone to please turn on their videos so I can actually see your faces, your reactions? Because that's the whole point. I want to talk to actual humans. Nara, thank you so much for turning on your video. Hi, Karimi. Hi, Federica. Great to see you.
I feel your pain.
Thank you. So I wish we were here all in the same room. I can see your reactions. I can see you nodding, being puzzled, etc. and I'm going to be introducing a lot of terminology. So, seeing your reactions would be very helpful, and seeing your smile, Ismail, is really, really awesome. So thank you. Really appreciate it. All right. So throughout human history, epidemiology has been correlational. We basically say, oh, this person is going to Starbucks to buy a $7 coffee 5 times a week. They also have better health outcomes. Is it that Starbucks coffee is really that good for your health? Or is it that the people who can afford better healthcare can also afford to have a coffee every day, and therefore there's a correlation that's predictive of your socioeconomic status, but not necessarily causative? And sort of forcing everybody to have Starbucks every day would not necessarily lead to better health outcomes. And that's why causality is important. And that's the promise of genetics. The promise of genetics is that these genetic associations are causal. Because if I am obese and I have inherited these alleles, I haven't inherited these alleles because I'm obese. It's the other way around. I am obese because I have inherited these alleles. So that basically tells us that it's very unlikely that somehow my genome changed with these common variants from my parents after I decided to eat a lot or to not exercise. So, and by the way, I am homozygous risk for the strongest genetic association with obesity. That's why I haven't eaten, I don't eat until noon. And I go beat myself at the gym every day after biking, after rowing, and after lifting weights. So anyway, that's sort of, the situation here, that it's not causation, that it's not correlation, it's really causation. So that's the promise of genetics. It gives us the path to causality. It gives us the ability to now look within the genetic loci and say, what is the mechanism? And if I understand that mechanism, then I can truly go and intervene. And what are the new target genes that I might be able to now build therapeutics for, and ultimately enable personalized medicine and precision medicine? So that's the promise of genetics. So what is the challenge? The challenge is, if you look within these genetic loci, you basically see that there isn't just one genetic variant, but there's a dozen, or in this particular case, 89 genetic variants that are all co-inherited and co-associated with disease. And the reason is these haplotype blocks that I mentioned earlier, that basically there's recombination hotspots here, but most of the time when a mother or a father gives part of their chromosome to their child, they give that entire chunk or none of that chunk. Basically, that entire allele gets a risk or no risk. You either get all of the variants or none of the variants that are associated with obesity. And that's why genetically we can't distinguish them. And it's very difficult to sort of recognize what is the causal variant. But much more importantly, if you look within this locus, and if you look at 93% of the genetic loci associated with complex traits, what you see is that none of them actually perturb the protein. If you look at this FTO gene, which was named fat and obesity associated after this association was discovered, this gene has exon 1 somewhere around here, exon 2, that's the protein coding component. And if you squint at the slide, you'll see this tiny little dot exactly where my pointer is. That's the exon, that's the protein coding portion. And that's the protein coding portion again. The rest is non-coding. And in this particular genetic region, none of the variants perturb the protein. And in fact, in 93% of cases, none of the variants perturb the protein. So that basically means that we can't just simply say, aha, it's the FTO gene. We have to say, we don't know what the target gene is. And in our own research in the New England Journal of Medicine a few years ago, we basically showed that the true target gene is actually sitting 1.2 million nucleotides away, several genes over. So that basically tells us that we can't simply look under the lamppost where the genetic association is. So a lot of geneticists basically say, aha, there's 5 genes here, it's one of those 5. And when there's a single gene, they say, great, we only have 1 gene. And I was introduced as the speaker of as the author of the most depressing paper of 2015 in an industry alliance collaboration, because I basically, my lab showed that this association, even when you have a single gene in that locus, it might not even be that gene. And industry had spent probably upward of $1 billion in trying to understand the function of FTO and in trying to target it therapeutically, and everything fell short. And the reason is that they were after the wrong gene. So that's the challenge. The challenge is that the vast majority is non-coding, the target gene is not known, the causal variant is not known, and that also means that the cell type of action is not known. So FTO contributes to obesity. How? Does it change my adipocytes? Does it change my brain? Does it change my muscle? Does it change my liver? Does it change my volition to exercise or to eat sweet? Does it change my taste buds? So what cell type in my body is responsible for this association? We don't know. What are the relevant pathways? How do we start beginning this therapeutic development program if all we have is a region and we don't even have a gene or a cell type or a mechanism or a pathway? That's what my research is all about. And that's, in my view, one of the greatest challenges of our generation, being able to turn these extraordinary insights of hundreds of thousands of genetic loci into mechanisms, into circuits, into knobs that we can use to intervene and reverse disease. And the way that we do that in my lab is through genetics, massive profiling of RNA and epigenome, data integration, and then dissemination. So with genetics, we start with these genome-wide association studies, showing you a different example for Alzheimer's in this particular case. So ApoE, apolipoprotein E, is the strongest association with Alzheimer's, and it has two alleles, ApoE4, which is the risk allele, and ApoE3, which is the protective allele. And carriers of ApoE4 have 10 times higher risk for Alzheimer's. Carriers of FTO risk have 1 standard deviation more weight, so much more likely to be obese. Like basically you switch your BMI suddenly with 7 pounds as an adult of excess weight. And ApoE4, 10 times higher risk. So the question is, how are these genetic variants acting? So we start with common and rare variant associations and the regions that are associated with disease. We then profile RNA expression and the activity of the epigenome in healthy and in disease samples. And the reason for that is that we want to understand how is the genetic variant acting. And we want to take these genetic variants, take the carriers of ApoE4 or the carriers of ApoE3, and then ask what is different at the molecular level within the cells of these individuals. Okay? So is this locus, for example, leading higher expression of the nearby genes? Or is it leading to sort of higher accessibility? You know, how is it manifesting at the cellular level, at the organ level, and so on and so forth? So we profile RNA expression, and our RNA expression everybody understands, but we also profile the epigenome. So what is the epigenome? DNA inside your cells forms this beautiful double helical structure, which is the basis of inheritance. And then this DNA is not just swimming around naked inside your cells. What it's doing is that it's wrapped around these nucleosomes. These are capturing 144 nucleotides around the nucleosome and about a 50 nucleotide linker, and that can be variable. So you can think of it as 200 nucleotide chunks, 200 nucleotide packets in the genome that are basically colored according to their function in that cell type. So as you know, you have trillions of cells, and every one of your cells has exactly the same DNA. We have hundreds of different cell types in our brain alone, and thousands across our body. Every one of them has a different phenotype. My neurons, for example, that span from my head all the way to my toes, are very different than the cells of my nails or my hair or my cardiac cells or my retina cells, and so on and so forth. So basically, this extraordinary diversity of the human immune system, the human nervous system, of the human skin, of every aspect of human physiology, all of that comes from epigenomics, from the ability to remember what subset of regions are important in every cell type. And the way you remember that is by giving different colors to these packages, so that in my liver cells, I will turn on all of the packages that have the yellow color there, and in my brain cells, I will turn on all of the packages that have the yellow color there. But then these are established in different locations of the genome, enabling us to have a single book of life with these 23 chromosomes. But every profession, every plumber or nuclear physicist has different chapters of that book of life that it opens up and reads. So in the same way that all of human knowledge is out there on Wikipedia, but you can sort of read different subsets depending on your profession, our genome has all of the knowledge inside the nucleus, but it only remembers the subset of chapters that are responsible for its own profession. And this is encoded through the histone proteins that make up nucleosomes. So every nucleosome has 8 histone proteins, H2A, H2B, H3, H4 in the vast majority of cases. And those have long amino acid tails, which are post-translationally modified with modifications that then allow readers and writers of chromatin to be able to sort of turn on and turn off these particular regions. Okay, can you guys give me a thumbs up based on awesome, great, good. So that's basically the core of epigenomics. Epigenomics is basically the modifications that the DNA can undergo that allow different cell types to have different functions. And the 3 types of epigenomic modifications that are primary are DNA methylation directly on the DNA on CpG dinucleotides, histone modifications, which impact about 200 nucleotides at a time, and then DNA accessibility, which basically tell you whether the regulators that are responsible for turning genes on and off are able to bind or not in a condensed chromatin or in an accessible chromatin. Okay, so again, you can scan any of these QR codes to basically read a lot more about the various papers that our lab has written on these topics. So we profile RNA and epigenomic variation, not just between cell types, but also between individuals, to understand how is genetic variation impacting these molecular phenotypes in healthy individuals, to understand the impact of genetic variation, and in disease individuals to understand the combined impact of genetics and environment in disease. We then integrate all of these data sets computationally to predict what are the driver genes, regions, and cell types. And then we validate these predictions through human cells and mouse models. So I'll basically give you examples of what are the extraordinary things that we can learn using this approach: genetics, profiling of RNA and epigenomics, integrating the data, and then validating the predictions. So I'll make a tiny pause here. I'll be right back. Okay, I'm back. So let's dive right in. So what do I mean by circuitry, first of all? When I say we want to understand the circuitry of human disease in my first slide, what I mean by circuitry is that every locus in the genome that is associated with disease has very specific variants that are driving this association. We heard before from Nara's presentation that it's not just the gene, it's the specific variants. It's not just a locus, it's the specific nucleotide. And now we want to understand, how are these nucleotides connected in these circuits? How do we find the downstream target gene? How do we find the upstream regulator? So this is how we find them. We basically start with the specific genetic variants, and then we ask, number one, where are these variants sitting in the genome? What are the annotations of these regions? And that's where the epigenomics comes in. So basically, we have developed methods for understanding the language of the epigenome, understanding where are these yellow tags and where are these red tags and these green tags, and systematically be able to identify regulatory control regions. So I was the senior author of the Roadmap Epigenomics paper in Nature that was basically an issue of 20 different papers through dozens of collaborations across many, like dozens of institutions, where we basically laid out the framework for understanding the human epigenome, for like annotating the human epigenome. And the output of that was basically a series of annotations that basically tell us in liver, in stem cells, in T cells, in placenta, what are the active control regions for different genes. And understanding where are these genetic variants sitting is extremely important for that. The second question is, what cell type are they active in? So basically, number one, where are the control regions? Number two, what cell type are they active in? Number three, what are the specific regulatory motifs that are responsible for these associations? So again, we've built resources that allow us to understand these motifs de novo. So we wrote a series of Nature and other papers that basically provided the first catalogs of regulatory motifs in the human genome through evolutionary conservation, comparative genomics, and other methodologies. The third is, who are the regulators responsible for binding these regions? And again, I was one of the senior authors of the ENCODE papers that are basically looking at how we can systematically understand all of the regions that are bound by a particular regulator. So if we take MEF2A or TCF4 or FOXO3 or GATA3, we can basically carry out a chromatin immunoprecipitation experiment. That's basically pulling down with an antibody all of the regions that are bound by a particular regulator, cutting up these regions, and then sequencing them to figure out what are all of the loci in the genome that were bound by a particular regulator. So as part of ENCODE, we wrote, of the Encyclopedia of DNA Elements, we wrote a series of papers outlining all of the binding sites for different regulators and how these are connected. And then again, as part of the Roadmap Epigenomics and ENCODE project and EpiMap that we recently published, so you can click on that here, 2 years ago in Nature, we basically now also painted the circuitry, the connectivity of which enhancers and promoters turn on coordinated fashion. And that allows us to now say that this enhancer targets this gene, that enhancer targets that gene, and so on and so forth. Okay? So what I mean by circuitry is I don't want just a region of association. I want the specific genetic variants, the specific regulatory control regions, the specific cell types where these regions are active, the regulators that are binding them, the motifs through which they're binding, and the links to the downstream target genes. Okay? Can I get a thumbs up as to who's with me now? Awesome. Great. Beautiful. Good. And even those with your video off, please do, give me a thumbs up when you can. Okay, hold on one second. My water bottle is in. So let's go back now to the 2 slides that I showed you earlier. First, I showed you a genome-wide association for FTO, for obesity, and then I showed you one for Alzheimer's. Let's see how our methods apply to those associations. So we basically said, okay, listen, I'm homozygous race for FTO, for obesity. I'd like to know how it works. So we established this absolutely wonderful collaboration with Melina Klausnitzer, Hans Hauner, and many other folks across the world to basically try and dissect what I like to joke, a single bit of information in the human genome, a yes/no answer. If you have a T or a C at that location, how does it actually function? And that's where all of this circuitry and all of these resources that I mentioned earlier come in. So through the motif analysis that was led by Melina, we basically asked, are there different motifs conserved in this locus? So basically what we found is that there is a single motif among these 89 common variants that are in this region that I showed you earlier. So among these 89 common variants, one of them, RS1421085, disrupts an evolutionarily conserved motif for this AT-rich interacting domain factor ARID5B. So I'm a carrier of AAC-ATT. Most of you are carriers of AAT-ATT. So in your chromosomes, ARID5B can bind, and it binds this 12,000 nucleotide super enhancer. This is basically not just a control switch on your wall. This is the master, circuitry board in your basement that basically controls the developmental fate of your adipocytes. So what we showed is that there's a 12,000 nucleotide adipocyte, pre-adipocyte enhancer that basically is controlled by the binding of this regulator. When this regulator can bind, it shuts off that enhancer. When the regulator cannot bind, that enhancer is overactive, and that's like 12,000, nucleotide master controller. And what that controller does is that it overexpresses IRX3 and IRX5. How far are those genes? IRX5 sits 1.2 million nucleotides away from this variant. And IRX3 sits 500,000 nucleotides away from this variant. So there's several genes over. And what this basically tells us is that you can't just look under the lamppost. It's not just the FTO gene. Notice the FTO gene itself doesn't feature anywhere here. It's the variant inside that intron of FTO that is responsible for the association. So through a series of elegant experiments, collaboration with this extraordinary team, we basically showed that when IRX3 and IRX5 get overexpressed, they shut down thermogenesis, which is the generation of heat. And that's the mechanism through which your adipocytes are able to burn calories. So when you consume a high-calorie diet and you don't burn all these calories through your day, every single one of these calories is going to be stored in your adipocytes. And that's why I'm at the gym. And that's why I don't have, I cannot eat that much. So that basically means that all of my calories will be stored, not burned. But you have the ability to burn these calories or to store them. And again, when I say you know who you are if you have to constantly exercise and watch your diet. So, this is like, we all know our metabolism. And again, this is polygenic. This is by far the strongest association, but there are many, many other contributors. So we basically now have a circuitry for understanding and basically saying it's not just, oh, you got unlucky. It's here's the mechanism through which you got unlucky. But why are we excited about this? Because by understanding the mechanism, we can intervene. We can basically say, can I increase or decrease the expression of the upstream gene? And can I increase or decrease the activity of the downstream targets? Can I go in CRISPR-Cas9 experiments and edit a single letter in that genome to establish the causality of that association? And we carried out these experiments, and in every one of these cases, we showed that we could switch back and forth between lean and obese phenotypes, between thermogenic and lipogenic states. To give you one such example, we took primary adipocytes from obesity risk individuals, homozygous carriers like myself. And we basically took these adipocytes out of the body and we edited, using CRISPR-Cas9, a single letter from C to T to restore the AT-rich motif that's bound by AT-rich interacting domain factor 5B. And what we showed is that in the basal condition and the isoproterenol stimulated conditions, we can basically measure thermogenesis as oxygen consumption rate, uncoupled as a percent of basal. And what we can see is that you can restore, with a single letter change, you can restore thermogenesis by a factor of 7 in the control and in the stimulated condition. This is a big deal. Basically says that, I don't know how much more precision medicine you can get from 3 billion nucleotides to pick a single letter and edit that letter and show that you can restore the phenotype. So that's very promising. It basically says that this is the causal variant and that this is the causal mechanism. And then we went into the downstream target genes, IRX3 and IRX5, and we said, what if we manipulated those as a target against obesity? And we took normal mice, control mice, not obese mice, control mice. And we said, what if I shut down this gene that gets overexpressed in the obesity risk individuals? And what we found is that IRX3, effectively, when you overexpress it, will turn your fat cells extremely thermogenic. So these mice lost 50% of their weight under this adipose promoter with the dominant negative construct for IRX3. And when you put mice on a high-fat diet, normal mice gain weight in a high-fat diet. These mice are simply unable to gain weight. They did not change their exercise. They did not change their appetite. They just simply burned calories when they were awake and when they were sleeping. So if you guys are excited about this and would like some pills, please let me know. I have them. So this is preclinical. This is basically saying that in human cells and in animal models, we can basically take a genetic association, turn it into a circuit, manipulate that circuit, and reverse the disease phenotypes. And that's what we want to do systematically for every genetic locus in the genome. We want to provide the tools, and we want to provide the circuits, and we want to even provide the therapeutics to be able to reverse disease phenotypes. So this is one example, and this is another example. Looking at ApoE4, we took ApoE3 individuals, ApoE4 individuals, and ApoE3/4 individuals. So basically, these are homozygous risk, homozygous non-risk, and heterozygous individuals. We took male and female individuals, and we took Alzheimer's and non-Alzheimer's individuals, because we care not just about disease, but the mechanism of disease. And therefore, we want to know, even if you're not a case, even if you haven't been diagnosed with Alzheimer's, maybe because you died before that, if you're a carrier of ApoE4, what is the consequence in your cells? And we carried out single cell profiling of the brain of post-mortem donors in each of those categories. And we asked what are the changes that are happening in the 6 major cell types in the brain: excitatory neurons, inhibitory neurons, astrocytes, microglia, oligodendrocytes, and oligodendrocyte progenitor cell. And what we found is that there were changes in all of the cell types, but dramatically, cholesterol biosynthesis was greatly altered in oligodendrocytes, specifically in the ApoE4 individuals, whether or not they were cases or controls. And through a series of elegant experiments by Joel Blanchard, Leila Akai, and other members of our team in collaboration with Li Hui Tsai at the Picker Institute, we basically showed that in the ApoE4 risk individuals, the cholesterol that was meant to myelinate neuronal axons was not being transported properly. That cholesterol biosynthesis was dysregulated and cholesterol transport was dysregulated. And when we looked at ApoE4 individuals, we basically showed that cholesterol was accumulating in the endoplasmic reticulum of oligodendrocytes, and that the myelination, these fat coating of these axons, was greatly reduced and sometimes completely absent. So we basically said, great, why don't we restore cholesterol transport using cyclodextrin or any other chemical that can allow this kind of facilitate this kind of transport? And what we found is that in human cells, the lack of myelination was basically recapitulating ApoE4 oligodendrocytes, and it was restored by cyclodextrin. And in mice that had lost their memory, cyclodextrin restored myelination, and it also restored cognition. So again, we go from a region of association to a circuitry and ultimately a therapeutic. And that's what we want to do systematically. So in the remaining time, what I want to do is highlight the different parts of making this possible. How do we build these reference epigenomes and use them to predict disease-relevant tissues? How do we understand the impact of genetic, epigenetic, and transcriptional variation in the context of disease? How do we understand this at single cell resolution? How do we understand cell type-specific genetic effects? How do we understand these impacts across multiple phenotypes and across multiple tissues? And how do we build these perturbations in very high throughput? Okay? So to build these epigenome references, we basically worked very closely with a Roadmap Epigenomic Consortium and with the ENCODE Consortium, and now currently with the International Human Epigenome Consortium, IHEC. And in those collaborations, we basically had many institutions that were providing samples of different regions of the body and different regions of the brain and different stages of development and different primary cells and different in vitro differentiated cells. And for each of those, we profiled these histone modifications, open chromatin, DNA methylation, and gene expression. And we were able to understand now the language of the epigenome by building computational methodologies that used multivariate hidden Markov models to scan the entire genome across hundreds of cell types and tissue types, and basically ask what are the modifications that are co-occurring, and can we now understand these unique signatures, and from those signatures infer the function of these different types of combinations. And we defined enhancer regions that act at a distance, promoter regions that are acting, exactly where the transcription starts, transcribed regions when genes are active, repressed regions, using these histomodification marks. So we had a series of papers in Nature Biotech, Nature Methods, and Nature that basically established this chromatin states methodology and built tools for understanding this epigenome. So we could now go and annotate the epigenome of these hundreds of cell types, and for the first time correlate the genetic variants that are associated with dozens of different disorders and the enhancers that are active in dozens of different tissues. And the result was striking. So what we found is that the genetic variants associated with height are very specifically enriched, colocalized in enhancers that are active in embryonic stem cells. Genetic variants associated with immune traits like type 1 diabetes, allergies, and so on and so forth, were associated with enhancers active in T cells and B cells. Genetic variants associated with blood pressure or heart repolarization intervals were very specifically localizing in the left ventricle of the heart. And this is where the blood pressure gets built up before being pumped in the rest of the body. Notice here, I'm not even using genes. Proteins are not even featured here. This is just non-coding variants sitting in annotations of non-coding regions. So we can directly compare the epigenome annotations and the gene and the genetic correlation. And now we can say that these genetic variants are probably acting in the left ventricle. Again, that's the part where the blood pressure gets built up before you pump it in the rest of the body. So it makes sense that these genetic alterations are in fact somehow impacting the left ventricle. If you look at fasting glucose-related traits, as we know, associated with type 2 diabetes, what you see is very specifically pancreatic islets. That's where the enhancers that are enriched in the genetic variants are sitting. If you look at cholesterol, those genetic variants are sitting in enhancers active in liver. So we can start making some bets here. Inflammatory bowel disease. Inflammatory, I would expect immune cells. Bingo. Bowel disease, I would expect digestive tissues. Bingo. So anybody wants to play some bets for Alzheimer's disease? So I would have put all of my money in brain, and then look, no, unfortunately, it's not brain. So what's going on here? Alzheimer's disease variants, you would expect them to enrich in brain, but they don't. What's going on? What's going on is that they're enriched in very specifically CD14 plus monocytes. So what's CD14? It is the mark of circulating monocytes and tissue-resident macrophages. These are the immune cells of our circulating body and of our diverse different tissues. And in particular, the macrophages or the immune cells of the brain are known as microglia. So we basically carried out an experiment where we sorted cells for PU.1 or SPI1, a regulator associated with microglia, and NUN associated with neurons. And we found that if we carry out these epigenomic experiments separately in neurons versus microglia, the genetic enrichment using LD score regression for linkage equilibrium score regression, the enrichment of the genetic variants was very specific to the immune cells. And what we also found is that if you carry out experiments in mice using the CKP25 mouse model that Li Huitai at the Picard Institute developed, you basically see that immune changes are happening much earlier than neuronal changes, suggesting, in fact, that inflammation and immune dysregulation might be a causal component of Alzheimer's disease. So we wrote this paper in Nature in 2015, basically saying that the conserved epigenomic signals in both mice and humans reveal an immune basis of Alzheimer's disease. So that's extremely important. Basically tells us that, if you want to address new therapeutics, you should be really looking at the causality of where these variants are acting. So we expanded from this Nature paper in 2015 to this Nature paper in 2021, from 127 epigenomes to 834 epigenomes. From 54 enriched traits, we went to more than 200 enriched traits. And we basically were able to now start mapping diverse traits to the tissues of action. So if you look at educational attainment, intelligence, cognitive performance, it localizes very specifically in brain. And that's because the vast majority of that is excitatory and inhibitory neurons. And so is, unfortunately, depression, neuroticism, schizophrenia, and so on and so forth. If you look at filtration, you see kidney. If you look at Alzheimer's, you basically see immune cells. If you look at cholesterol, you see liver, and so on and so forth. And you also see some traits here in the middle, which are localizing not just in one tissue, but in many tissues. Every circle here is a pie chart that basically tells us what is the relative contribution to those enrichments of different tissues in the body. And coronary artery disease was, in fact, one of the most complex. If you look at QT interval, heart rate polarization, very specifically the heart. Educational attainment, very specifically the brain. Alzheimer's, mostly immune, some brain. Waist to hip ratio, BMI, adipocytes, muscle, kidney, it starts getting more complex. Health span, adipose, digestive, epithelial, blood, fibroblast, intestine, lung, many different tissues, embryonic stem cells. And if you look at coronary artery disease, you basically have 20 different tissues that are enriched in very different parts of these tree, relating the different tissues to each other. So that basically means that perhaps we can use the epigenomics to start partitioning the complexity of those traits. And in particular, to look at the subset of genetic variants that are associated with coronary artery disease that are acting in liver versus the subset that are acting in coronary artery versus the subset that are acting in thyroid or adipose, and so on and so forth. And if you look at the biological enrichments of the corresponding genes that are proximal to that subset versus this subset versus that subset of genetic variants, then you see dramatically different enrichments, suggesting that we can now take these complex traits and indeed partition them into lipid metabolism, into epithelial proliferation, lipid homeostasis, efflux of phospholipids, and so on and so forth, associated with the different tissues that are being involved. This is very exciting. Basically says that we don't have to think of disease as a monolithic disorder. We can start asking for personalized medicine. What are the parts that every single one of these patients is at increased risk for or increased dysregulation for? And we can use this to now go back into these genome-wide association study hits and start asking about the subset of variants that are sitting in coronary artery or in liver, and so on and so forth. And as you go down this list, you basically see that different variants are associated with different subsets of this function. And we find some variants such as PCSK9, which appear to be acting through a liver mechanism, and eDNRA through a heart mechanism. And PLPP3, basically see these genetic variants here are linked to two different genes, one expressed in the heart, one expressed in the liver, suggesting that some of these genetic loci might be multigenic and multi-tissue in their function. They have many ways of acting. Plio means many, tropos means ways, many ways of acting. And sort of this suggests both multi-gene and multi-tissue pleiotropy. So up until now, I've been basically annotating these reference epigenomes to predict the disease-relevant tissues, but we can also use comparative genomics to annotate these genomes. As I mentioned earlier, we can use evolutionary conservation of regulatory motifs to understand which of these letters might be more important or less important. And we've developed a series of evolutionary signatures through, again, these series of papers in Nature, in yeast, 4 mammals, 12 flies, 29 mammals, and recently 200 mammals in a series of papers that were just published a few days ago. to basically systematically annotate where are the protein coding regions, where are the regulatory RNAs, the microRNAs, the regulatory motifs, and so on and so forth, through their patterns of evolution. And using this, we can go and revisit non-coding GWAS loci. So there were 118 seemingly non-coding loci where there was not a single protein coding variant, protein altering variant. But in fact, what we saw is that when we annotate our new exons that we discovered through these epigenomic signatures, we can now recognize that these locus, for example, associated with myopia, we were somehow blind to the fact that there's a new exon here that's specifically associated with retina and is expressed in the retina. So that suggests new mechanisms for protein altering functions for these seemingly non-coding genetic associations. And again, I mentioned earlier how we can use this evolutionary conservation to recognize motifs and to discover a dictionary of motifs de novo. And we can also use that to understand much smaller genomes, such as the SARS-CoV-2 genome of COVID-19. And then we use that to show that a protein coding gene that was annotated, namely ORF10, is in fact a non-coding RNA, doesn't evolve at all like a gene, and that there's a new gene here, ORF3C, which is overlapping ORF3A. So we're able to discover new genes, interpret mutations, and so on and so forth. So that's about understanding the reference annotation of the genome, understanding the genes, the control regions, the motifs, and the circuitry. We now want to start asking how are these changing between individuals? And in particular, we want to bridge the gap between genetic variation and disease. Instead of just saying, this genetic variant here is acting all the way to Alzheimer's, I want to say that, oh no, it's acting in the brain. It's affecting an enhancer, which is then changing the expression of a gene, which is affecting amyloid beta clearance and ultimately Alzheimer's. And the challenge here is that many of those arrows that are unidirectional when they come out of the genetics are in fact bidirectional in these intermediate phenotypes because of feedback from the environment or because, feedback from the disease itself. And in particular, some of these genes that might be up in Alzheimer's might not be causing Alzheimer's. They might be fighting against Alzheimer's. So to do this, we basically looked at 700 individuals that had donated their brain to science. And we looked at the dorsolateral prefrontal cortex, and we looked at the methylation patterns of those. And then we asked, how is the genetics, the transcriptomics, the epigenomics, and the phenotype relating to each other? And we found 50,000 loci where the genetics and the epigenomics were highly correlated, suggesting that if I know the genetics of that individual at conception, I can predict their epigenome at 93 years of age in an inaccessible tissue just based on the correlation between genetics and epigenomics. And I can do this for 50,000 loci across the genome. And for every one of those loci, I can now ask not just how is genetics associated with disease, but how's genetics predicting methylation? How's methylation correlated with disease? And how is the genetic component of methylation correlated with disease? And if I just look at all of methylation, it's a bidirectional association. But if you look at just the genetic component, it's a unidirectional association. And the reason for that is that this is not all of methylation. This is just a subset that I can predict genetically at birth. And that subset is not a consequence of disease. It's only a cause of disease when the two are correlated. So we can use Mendelian randomization and other methods to start inferring causality of these intermediate time points, to start predicting new loci, and to start going into existing loci here in purple and predict driver genes and whether they're positively or negatively correlated with Alzheimer's, and also predict new genes in loci that are not genome-wide significant. And I can do that through methylation or through transcription to understand the causality here. And everything I've shown you so far has been at the tissue level resolution, but we can do that at single cell resolution. So we wrote a series of grants to basically study single cell changes in disease across thousands of human brain and other samples in Alzheimer's, in frontotemporal dementia, Lewy body dementia, amyotrophic lateral sclerosis or ALS, a muscle disorder, Huntington's, psychosis in Alzheimer's, schizophrenia, bipolar disorder, Down syndrome, post-traumatic stress disorder, autism, addiction, aging, cardiac disease, obesity, and exercise. And for each of those, we gathered cohorts of patients working with hospitals, and we looked at dozens of cell types, multiple regions of the body and of the brain, and then looking at RNA expression and DNA accessibility. In the first paper that we published in Nature a few years ago, we basically looked at 48 individuals, 24 cases, 24 controls at different stages of Alzheimer's progression. You can see here cognition being high and eventually dropping off, and then pathological variables being low and eventually increasing, such as amyloid, neurofibrillary tangles, and other pathologies. We found that early changes in Alzheimer's were cell type specific. Late changes were very cell type shared. We found that 3,000 genes were differentially expressed between men and women prior to AD, but with AD, 6,000 genes were differentially expressed. And we found a lot of sex-specific and cell type-specific changes. In particular, we found that myelination was differently affected in men and women. And indeed, this was recapitulated with an increased white matter loss from the imaging studies of female individuals that was not previously reported and was a direct consequence of our transcriptional signatures. We have since expanded from 84,000 cells to 1.6 million cells, and from one brain region, the prefrontal cortex, to 7 brain regions, mid-temporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body. And we now have this extraordinary diversity of neuronal subtypes, glial subtypes in these different brain regions. For excitatory neurons alone, we have 30 different subtypes. For inhibitory, 23 different subtypes. And we can now see how dramatically these change between different brain regions. And we can actually sort all of our cells, our 1.6 million cells, based on the predicted transcriptional signatures that are associated with Alzheimer's, to basically find early stage and late stage cells, and then ask, where is each of these genes that is causally associated with Alzheimer's through genetics? Where is this change in expression across this progression? The single cell data allowed us to also recognize anatomical differences in where these cells are positioned. If you look at the hippocampal formation here between dentate gyrus, CA1, 2, 3, subiculum, and these different layers of the entorhinal cortex, these cells are actually clustered differently based on their transcriptional profiles, allowing us to translate back and forth between anatomy and transcription. And we've now started using subcellular resolution spatial transcriptomic technologies to start understanding where in the body are different changes happening and where within these tissues. If you look at coronary artery disease, we basically looked at open heart surgery and ex vivo samples and looked at the expression of many of these genes that are changing at single cell resolution. And we found that they localize very specifically in the intima luminar media. And in particular, we found that these macrophages, again, that are now engulfing these lipids in obese individuals that are now having cardiovascular symptoms because their lipid stores are saturated and their lipids are now circulating the bloodstream. We found that these foam cells that we call macrophages that have engulfed these lipids are dramatically changing in a very specific spatial orientation. We've developed methods for translating between spatial and expression data. We've used these millions of cells to learn modules of genes that are co-active and co-expressed, and how these modules, even in rare cell types like microglia, are in fact revealing the coordinated changes that are happening associated with distinct phenotypic signatures of Alzheimer's. In schizophrenia, we're able to find master regulators responsible for these changes. We're able to predict schizophrenia cases versus controls based on their transcriptional signatures, not just in one cell type, but across dozens of different cell types. And we find that some individuals that are predicted to be schizophrenic are in fact controls. But in this particular case, the son of this person was in fact diagnosed with schizophrenia, suggesting that perhaps our transcriptional signatures are capturing something that might be missed in the diagnosis of these people. We now can go into dozens of genetic loci associated with schizophrenia and start predicting what are the candidate driver genes that are upregulated or downregulated, what cell type are they acting, and how they're linked in the folding of the chromatin. And again, predict these upstream regulators that are acting both in early development and in adult. In psychosis, which is psychotic episodes in Alzheimer's, we're finding not overactivation as you would expect with psychotic episodes, but instead repression of excitatory and inhibitory neurons, suggesting that in fact it's this repression rather than overactivation in psychosis in Alzheimer's. Looking at addiction, we're finding changes extremely specifically in opioid receptors in the nucleus accumbens that are painting nearly every gene involved in this AP1 family of regulators, suggesting again specific therapeutic endpoints that we can manipulate to combat addiction. In ALS, a motor disorder, and FTD, a frontal temporal dementia that is affecting the frontal cortex, is manifesting in psychiatric symptoms, we're finding that the same exact cell type is altered orthologously. These long-range projection neurons, known as von Economo cells in frontal cortex and BET cells in the motor cortex, are in fact orthologosly changing. In Huntington's disease, we're finding that direct pathway versus indirect pathway spanning projection neurons are losing their identity. In pathogen-associated Alzheimer's, we're finding dramatic changes in immune cells and in vascular cells, which are the entry points of the infection. In COVID, we're finding a dramatic increase in astrocytes for COVID plus individuals, and that there's dramatic changes in both microglia and, again, vasculature, and specifically endothelial cells where the blood oxygen exchange is happening. In addition to the cholesterol changes that I mentioned previously in ApoE4, we're finding that other lipid transporters are in fact dramatically altered, changing fatty acid metabolism and cholesterol regulation. We're able to distinguish different subtypes of microglial cells and predict their upstream regulators, and now use these regulators to start reversing inflammation and bringing back homeostasis. So inflammatory states of microglia turn out to be requiring many fewer regulators, as if they were closer to a default state. But the upstream regulators associated with homeostasis can be basically overexpressed to drive back homeostasis and prevent the damaging inflammatory states of microglia in Alzheimer's. Again, another Nature paper last year in vasculature, looking at the single cell dissection of vasculature. And just a week ago, we published a Nature Neuroscience paper, basically showing how that vasculature is changing between different regions of the brain and between different cases and controls in Alzheimer's disease. Looking at the epigenomic changes at single cell resolution, we can find coordinated changes in the accessibility of promoters and enhancers and in the expression of the corresponding genes, and use that to find upstream regulators in every cell type that are responsible for these changes. We're able to now go not just into the expression levels, but also in the sequence changes that are happening with somatic mutations in the transcriptome of these dysregulated and senescent cells, and show that these de-identified cells are in fact losing their identity and accumulating mutations, and that this is associated with this large-scale epigenome erosion that is happening in late-stage neurodegeneration, where the closed regions are opening up and the open regions are closing. And across multiple phenotypes, the electronic health records, we can now start integrating these information to start identifying hallmarks of Alzheimer's that are repeatedly altered across different individuals and that are associated with distinct phenotypic signatures, again, allowing us to now understand the risk of each individual and suggest personalized medicine interventions for those. We're able to look at specific interventions in the medical record and infer causality through the temporal relationships, and look longitudinally in cancer at the mutations that are accumulating in different lineages of the same patient when the cancer metastasizes. And here, looking at 100+ individuals at single cell resolution for immunotherapy response to melanoma, metastatic melanoma treatment, we're finding that there's a specific regulator upstream that would predict increased success. And when we go and manipulate that regulator in mice, we basically see that a combination immunotherapy and chemical interventions through these drugs can basically lead to dramatic eradication of these cancers. I mentioned earlier the effect of exercise and diet across multiple tissues. So we conducted a multi-tissue study across muscle to different types of adipose tissue, intestine, cortex, hippocampus, and hypothalamus, liver, heart, and blood for the impact of exercise and diet in both human and mouse. And we found this dramatic reprogramming of your fat cells and of your fat tissue into thermogenic tissue using these therapeutic interventions and these exercise interventions to be able to now recapitulate them therapeutically. And now we have dozens of predicted driver genes that we are trying to systematically manipulate. So we have developed these high throughput methods that allow us to test 7 million enhancer constructs in a single experiment by cutting them off the DNA and then inserting them into self-transcribing reporter assays. And we've developed these modular and programmable CRISPR-Cas9 and dCas9 based systems for deactivated Cas9 that allows to recruit activators or regulators and change the epigenome directly, or edit individual SNPs or individual promoters and target genes. And we're now collaborating with my former postdoc, Andreas Penning, who's now a professor at CMU, to basically now test thousands of these genetic variations in human constructs for enhancers, but in a mouse whole brain to understand how these are acting in the context of many other cell types. And lastly, we've developed this extraordinary collaboration with Brand Peteluta and Marika Zitnik to start developing new chemicals based on these target genes, using this AI-driven drug discovery engine to basically develop different therapeutic candidates, do high-throughput experiments, and then use all of these different modalities for chemical intervention and use geometric deep learning, language models, and self-supervised learning to understand the protein language of chromatin, of protein folding and of chemical folding, and use them both in the same embedding to start reasoning about new chemicals and synthesizing these chemicals systematically. So I could tell you about all of the wonderful papers that we've written, but I want to leave you with the realization that this is far from over. Disease is still reigning. We need to completely transform pharma. We need to enable personalized medicine and individualized medicine by understanding the hallmarks of disease, understanding how to manipulate them, manipulate each of these hallmarks, and then using combination treatments for every patient to effectively overcome this burden of disease. And this will require a coalition across computer science, biology, chemistry, biotech, finance, pharma, patients, and hospitals. So I'm extremely fortunate to be at the head of this extraordinary interdisciplinary team across experimental and computational colleagues, and then to work with these extraordinary groups of individuals. I'll stop there and see if there's any questions. Thank you.
So thank you very much, Manolis, for your energy and your wonderful talk. I'm really impressed by what you and your team are doing.
Thank you.
So we don't have that much time, so I just want to see if anyone has any question.
If you guys have questions, feel free to type them in the chat or just raise your hand and turn on your video.
For sure.
I just have one naive question.
Of course.
When you talk about the annotations of the non-coding variants, my question is that for every non-coding variant, is it possible to have an accurate annotation? For example, is it possible to say that this variant is located in the region of enhancers, for example, or repressors? And if so, for how many % of variants actually you can do that?
Yeah, absolutely. So if you go to comptbio.mit.edu / let's see, haploreg, it will basically take you to a website where if you type RS1421085, you can basically submit this, and it will tell you for that variant of interest, what are all of the annotations. So it'll basically tell you this variant here, RS1421085, that I just typed. But it will not just say, here's this one variant, because variants are not inherited in isolation. Instead, there's a whole linkage disequilibrium block. So you can see here the LD score. And then it'll tell you every enhancer, every DNA accessible site, every promoter, every motif that's altered, every GWAS hit. And if you hover over them, it'll basically tell you, what they're associated with. And here you can see that this is associated with obesity, for example, with, dietary micronutrient intake. And, you can see here ARID3A as the motif. And, that's the variant that I focused on earlier. So yeah, you can do this for every variant in the genome. Just type your favorite variant and it'll tell you everything that it overlaps with. Now, again, these are genome-wide annotations. The other resource that you can go to is EpiMap. And then here, you can basically go to in the interactive browser. And here you can, that's the code. You can basically go to GWAS enrichments and then, choose, I don't know, waist to hip ratio. And then you can see all of these diagrams that I showed you earlier. You can see the enrichment, and you can look at individual enhancers. And then you can look at individual links with downstream target genes. And you can even look at the locus visualization that I showed you earlier. And we have 30,000 of these for every single locus associated with every single disease.
What about if you find a novel variant that don't match with any of these regions?
Just 1 second, I'm typing the two compile /haploref. I typed the two resources in the link.
Thank you.
So if the variants are not common, then that's where deep learning comes in, and that's where these convolutional neural network models allow you to predict the impact of these individual variants. We haven't made those tools available systematically, but that's one of the things that we're working on.
Thank you.
Of course. I see a question from Fri10am asking, where do you see the future of AI in precision medicine in the near future? So the place where I see it is really this, is basically, instead of just, and I encourage you to click on these two links, because you can see two TEDx talks that I've given in 2010 and 2021 that are basically describing my vision for that future. But the idea is that we don't want to be surprised. We want to move from being surprised to prognosis. We want to move from misdiagnosis to better biomarkers and multimodal diagnosis. Instead of just treating the manifestation of the disease, we want to address the root causes and the hallmarks. Instead of thinking of disease monolithically, we want to understand the heterogeneity of symptoms and the heterogeneity of causes. And again, understand the components. You can take Alzheimer's, break it up into amyloid and tau and inflammation and lipids. Instead of thinking about silos, where every tissue is studied in a different lab and every department is studying a different tissue, we want to understand the interplay between these different tissues, the multi-tissue communication, the commonalities, the sharing. We found lipids underlying obesity, underlying Alzheimer's, underlying cardiovascular disease. We want to understand these commonalities and go after these hallmarks in a systematic fashion. And instead of just treating too late, we want personalized preventive interventions. And to do this, we need to understand polygenicity, how thousands of variants converge into a small number of pathways, understand the hallmarks and these causal pathways, reverse disease circuitry through these directly targeted manipulations, enable individualized treatment by combination of pathways, where every patient is a distinct combination, and understand the burden for every person of these pathway perturbations, and understand how to combine omics with medical records through genetics, epigenomics, transcriptomics, and disease. I hope that answers your question, Fri10am.
Okay, thank you again, Dr. Kellis. This is the end of our session for today. If you have any questions, please feel free to contact us. Thank you, everyone, and looking forward to seeing you next Friday.
Amir, thank you for putting together this wonderful resource. I'm really, really thrilled that you're doing this.
Thank you. Thank you very much.
Bye, everyone.